Oh, it's on here. Where we are now, stop the last class. Communication is read. Just read.
So, people get to the golf.
Look at it, Dory.
But I just.
Where we are from? This one?
Everybody has.
Oh. No, you're having so. After. Right, so we know this structure of the DNA. Right, we did structure of RNA as well, right?
Yeah.
Yep. All right. Already, I showed you all the structure of DNA as well, right? That's the one that's all.
The way I'm looking for.
Yes. You want to
Like tea kind of.
what is the outside of the T? You want to like this?
What class are you signing?
Is something like that, then?
Yeah.
Right.
Yes. Yeah.
I read your DNA. Sure.
Nice. That's what I'm trying to find.
Yeah. Right.
I love it.
Right. So now we can start DNA replication. Right, so when would before which process would the DNA have to replicate or be copied? When would you want your DNA to be copied? During mitosis or meiosis, right? So right before mitosis or meiosis, you want your DNA to replicate. Remember, mitosis is when you start with one cell and it produces how much daughter cells? So you start with one cell and you produce for mitosis, right? And meiosis is start with one and you produce four. Right? So right before cell division, we must replicate the DNA. We must copy it. Right? So we have to go through steps in DNA replication. Right? So let's say we have our DNA. Right? Let's say this is our DNA strand, right? Now we know what's in our DNA strand. What would we have what's in it? Right? So we have the nitrogenous bases.
From class. You sure?
Right? So this DNA goes to the cell cycle will be closed up. Right? You don't want your DNA to be open because that can cause mutations and changes in the genetic makeup. If you change that, you change the type of protein being formed. Okay, so the DNA is enclosed. When it's for this type of structure again, when you have a double helix, right? So it's enclosed in a double helix structure. During DNA replication, it cannot be closed. You need it to open up for it to come. Right? What you're doing is really copying the bases. Right? So let's say,
So you're recording this class right now?
yeah. Right? Let's say this is the 5' end, right? So which end would this be? The triple. So what would this end be?
2 prime,
3', and this end?
5 prime.
Let's go and close the end double helix. Which base pairs? Bottom. Okay, so if this in the end, if this is E, right? If this is E, what base would be on this side? T, right? T is complementary to A. If this is G, C, if this is U, right? So now that's your question, right? Right? U still can't be in here, right? That's the RNA base. So if this was T, A, C, G, that'll be complementary pieces, right? Now, in DNA replication, the first step, right? Step 1, you want this DNA to unwind. So these bases, remember, they form hydrogen bonds between each other. That's what this helix includes. You want to break those hydrogen bonds so the DNA strand will open. So what happens? A special enzyme called DNA helicase will be used. DNA helicase comes in and breaks these hydrogen bonds, and that allows the double helix to open. The DNA strand do you draw it down? Right, so the first step, a special enzyme called DNA helicase unzips the enzyme. Right, now does it unzip the entire enzyme or just a part of it? The entire enzyme. When you're making a copy of DNA, you want the entire sequence to be copied. So the DNA helicase comes in at one point and starts to unzip. Think of it like a zipper. Unzipping the DNA helicase.
3 prime, 5.
Yes. We get to that as well. Right? So DNA helicase causes unwinding by breaking the hydrogen bonds. The second step, so we do these steps into the enzyme, right? We have something called DNA polymerase. So what does this mean? This DNA, this enzyme is responsible for the actual copying of bases. So after it unwinds, so your DNA open up now, the DNA polymerase comes in and starts to copy the bases. So by giving it, so if that was C here, the complementary base will be G. That was T here, the complementary base will be E. So it starts to copy it. The DNA polymerase copies. What does it copy? Provides complementary. Bases. Okay, so think of this enzyme as like a copy of base one, right? It helps it to copy the genetic sequence, right? And step 3, and the third enzyme involved in this is DNA ligase. So if you're trying to think of an analogy, think of this as like glue. Right? So remember, after your DNA unzips, right? And then it starts to copy. You're no longer going to have one strand of DNA or one double helix. How many double helix would you have? You'd have 2. So it starts with one double helix. After you copy it and do some send up with 2. Right? Right? So after you end up with these 2 double helix, you want to glue it back together. You don't want the double helix to remain open. So to glue it back together, we use something called DNA ligase. So DNA ligase strips back of the DNA. Right, I'll show you all the diagrams just now, but I just wanted to go through the different types of enzymes first. Right, so for now, just take down the double helix and the three main steps. You're right, up back a little bit. Oh, this row, little. So you prefer biochemistry part of it?
No, she not completely topic, she completely much
Oh, I was in bio. Well, and you have markers? Oh, one of. It was working good, you know. But there's. Same time. No, it was working good and then it just stopped. And I think somebody's stealing. Oh, somebody.
Then we have like a collection of everyone methods. You see all that's photos of here, this.
Ocasio.
What is that? So,
There you go, see what it is, just number.
Huh?
And she asked what it was, so.
The word is repeated, but I don't want someone from the translate curbside and I close it. And as I knew.
That's unit two, yeah. Unit two is zero, so.
You know, please, yeah.
Yeah, it's just the first two chapters given part of, and anything other people. I don't know how, I just. It has less calculations, so it'd be,
It'll be easier for me.
it's more theory. But it's basically organic chemistry. Okay. It's not good, though.
Like my friend sent me some kind of thing like. Like reaction, yeah, I remember I saw that around. Yeah.
Yeah, that's reaction to food.
It's like different reactions, so yes.
It's like, what it's in, is
Like you memorize it. Fun listen. But I want to understand, but not. And then like that power.
Yeah, maybe to jump.
That's just me. Well, it's just good. What you looking at? When you teach cameras, like when you need for it's still a flash. Yeah, I say cameras, strategies, you know. Why? What is our first stuff?
Yeah, we're going to teach us this.
I just feel pain do it. It is about one between you and me. What is it?
Yeah, no, this is hydrogen one.
No, please, it.
Yes. So it's perfectly complementary to T, right? And you really pull it together, it's hydrogen bonds. So there's a hydrogen bond between E and T.
Okay.
Right, so let's look at step two, right? Diagram one. So the DNA helix causes DNA to unwind. And this will happen throughout the entire helix, right? But I just want to show you all one part. So after it unwinds, the stone. Right, so remember this will be 5 prime. If this is 5 prime, this would be 3 prime. If this is 3 prime, this would be 5 prime. Okay? So what will happen is DNA polymerase is going to come in. These bases are exposed. Right? What DNA polymerase is going to do is going to bring in the complementary bases and attach it to them. So they said, remember, these two are the original strands. Right? So if this was A from here, it is going to attach a complementary base, which would be T, right? Originally, this strand had T. So it's going to bring in a base that is complementary to that, which would be A. Okay? Same thing with our control. This strand had G. So a complementary base of C is going to come in. This was T. This was C. So the complementary basis is going to show you. G on both sides. T, C, A, G. And this is happening because of the presence of the enzyme DNA polymerase. Okay, this is gonna happen through again inside your sequence of the DNA, right? And again, it's only because of DNA polymerase, right? It has some more steps after this, but in step 3, like I said, then in step 3, I don't want your DNA to remain like that. Okay, so what you want to do now, you want to zip this back up. You also want to connect the bases to each other. Now, what would happen here? You're going to form a new strand there, and you're going to form a new strand here. You all see that, right? So this would be a completely new strand, this entire side, and this would be a completely new strand. Okay? So we end up with 2 separate DNA helices. This is called semi-conservative replication. It's semi-conservative because they look at this new DNA strand, right? It has one of the original strands, but one of the strands is new. Right? So one of the 2 is happy. So that's why I get semi-conservative. This strand as well. One of these strands was from the original DNA, and one is a completely new strand. Again, semi-conservative replication. Only half of the 2 is from the original DNA. Right? And why is this important? What does semi-conservative replication ensure? It ensures that the DNA sequence remains unchanged. Look at this DNA sequence here, AT. The original one is AT. AT, AT. GC, GC, GC. Right? You do not want to change your DNA. If there are any changes, you'll change type of protein produced. Right? But you'll change the cell structure and function. Okay? So semi-conservative replication ensures that little to no changes occur within the DNA sequence. Right? Right? So just take out this diagram for step 2, and I'll start pulling notes on it. Kind of long. Right? When you're revising, the main thing to remember, try to remember the main steps, and then you could build up on it, right? Because there's a lot to remember. Yeah, okay, at least on a good diagram or something, so. It's all good.
Yes, I was actually thinking that I know someone good.
When I do a good job, they tell me to. That's why most of my diagrams be like that.
So confident.
Yeah, it's really all your fault. I could do it, it's just.
I see they make a TikTok about it.
Mm.
He wanted to get skinny.
Oh, what's that?
I didn't think that. I was in form 5.
My phone vibes.
Your form class.
Anyways, I don't know how I get them.
Should I read?
No,
Yes.
not a release. Tell me, where can we learn Jordan?
No, I don't know. You might not figure it out.
I don't think.
Perfection.
I don't think they come back after we do them last week.
Really? It was our group thing, right?
We showed them it on TV. Every time.
Show them more.
The screenshot.
Screenshot?
Oh.
What is it? That's
Hmm?
like a good time,
Well, we pin it on Cameo. We say Cameo send us it.
Open it and then I sit down.
So you want them to tell us you?
I'm not that message or people stuff. But that's not thanks to the agreement, it's right.
Hmm.
Good shit, I'm gonna die.
Hold on. Taking in note, right? So again, it's under the same heading, TNA replication.
You mean? Yes.
So we write in DNA replication is DNA replication is before cell division. DNA replication is before cell division. And is described as before cell division and is described as semi-conservative and is described as semi-conservative. Because each new double helix, contains one of the original polypeptides, polynucleotides, one of the original polynucleotides contains one of the original polynucleotides from the original double helix. Full stop. And so again, they do it in steps, right? So you're going to skip a line and put step 1. Step 1. Helicase unwinds the double helix structure. Helicase unwinds the double helix structure by catalyzing the double helix, by catalyzing the hydrogen bonds. Between base pairs. Between base pairs. Full stop. Each chain each chain or strand, each chain or strand, each chain or strand can then be used. Each chain or strand can then be used for the production of a new complementary polynucleotide chain. Full stop. Right? So as the DNA helication, right? It'll start at a certain spot. When it starts at a certain spot, it will move along the entire genetic sequence or the entire DNA. So for example, if it starts here, you're not just gonna work here, you're gonna keep moving up. Right? And right behind DNA, helicase, like right behind it, you're gonna have DNA on. Right? So it doesn't need to completely unwind for this to complete. As the helicase moves along, the polymerase follows closely behind. Right? So skipping a line. No, this is step 2. Skipping a line. DNA polymerase follows closely. DNA polymerase follows closely behind helicase. DNA polymerase follows closely behind helicase. With the function of linking, DNA nucleotides, with the function of linking DNA nucleotides in a 5' to 3', 5' to 3' direction. Full stop. It catalyzes. Oh, Cheyenne wants to make a payment after.
Okay.
It catalyzes the continuous. Or after 5' to 3' direction, just been a full stop, right? I remember first say that. Right? So full stop. It catalyzes the continuous synthesis of one. It catalyzes the continuous synthesis of one polynucleotide chain. In a 5' to 3' direction. In a 5' to 3' direction. Until it reaches till it reaches a replication until it reaches a replication fork.
Fork.
Fork, F-O-R-K. Stop. At this fork, at this fork, polymerase, at this fork, polymerase moves to the opposite. At this fork, polymerase moves to the opposite chain, moves to the opposite chain. Moves to the opposite chain, where it forms complementary, base pairs. In a 5' to 3' direction, where it forms complementary base pairs in the 5' to 3' direction. Full stop. On this strand, right, on this strand, open brackets, lagging strand, so as we call it, on this strand, in brackets, lagging strand, close brackets, close brackets. The polynucleotide chain on this strand, the polynucleotide chain is discontinuous. The polynucleotide chain is discontinuous. Only about 1,000, only about 1,000 bases long, only about 1,000 bases long. Full stop. These short pieces are called Okazaki. The short pieces are called Okazaki fragments. Short pieces are called Okazaki fragments. Please note the spelling.
Okay.
Right?
I can't remember. I was too short with in something and at something, right? And when you in something, leave in the spot. Yes. There's so much excellent. That's gonna be so long.
That's what we do. That was last week.
I studied MR.
Yeah, we didn't get to this. Right? Okay, no, before we move on, right? When I was talking about the replication fork, is like this is the area I'm talking about. So remember, the ending polymerase moves in one direction. 5' to 3', right? So let's say Well, this is upside down. In this one, let's just say it starts from here, right? So it will go from a 5 prime to a 3 prime direction, right? So this strand going in this direction. We call this the leading strand. Okay? Now DNA polymerase only works on the one strand at a time. Right? So it move it up, it get the complementary bases. After it reaches this kind of fork, this replication fork area, where the rest hasn't unwind yet. After it reaches the replication fork, the DNA polymerase is going to come off of this leading strand and they're going to start to work on the lagging strand. Again, it has to be in a 5 prime to 3 prime direction. So it jumps onto this side and then it starts to move downwards. These complementary bases will be there as well. Only as the DNA polymerase starts to move down, okay, then we'll lag in. When it starts to move down, the complementary bases will be attached here. Now the complementary bases that are attached here form a discontinuous strand, right? I can't draw 1,000 bases, so I'm not going to do that. Right, so it will be discontinuous, meaning they wouldn't be connected to each other. Okay? These discontinuous base strands are called the Okazaki fragments. Eventually, it will copy out the entire of the ligand strand, will come back up to the replication fork, and then it will continue along the rest of the DNA. Okay? Now, it's very important we understand which is the leader of the ligand strand. The leading strand goes towards the replication fork. The lagging strand copies away from the replication. That's how you'll identify. Right? We're missing anything.
So these leading strand, right? So we start at 5 prime, right? But if we start at 5 prime on that side, we go around 5 prime and on 3.
Right.
And if the other way comes to where,
This one?
yeah.
Right, so it can't be this way, right? Because if it can't start on this side, because here it's gonna wind this tunnel, right? So just look for where the replication fork is, and then you know which side to start. Right? Everybody good so far, right? Right. Right, now step 3. Right, so now we have to step 3. DNA ligase. DNA ligase is used. DNA ligase is used DNA ligase is used to join the Okazaki fragments. DNA ligase is used to join the Okazaki fragments. To form a continuous polynucleotide chain. To form a continuous polynucleotide chain. Full stop. Semi-conservative replication. Semi-conservative replication ensures that the original DNA is maintained every time replication occurs. And the original DNA is maintained every time replication occurs. Full stop. Right, so you're supposed to end up with after step, you have DNA one. Supposed to end up with this. Right? Two. Right? If you start with one strand, this will end up with two identical double helix or double helixes. Right? That's after step. Right? I just drawn it dotted, right? So the dotted one represented, okay, one of it is from the original DNA, and the other strand is a new one. So we can see how it fits into. Right, so we have to know an experiment or we have to know of at least one experiment to prove that DNA replication is semi-conservative, right? So what some scientists did in 1957, right? If you're all interested in science, this is Mesalon and Stahl, right? Your research among the time. They used E. coli, right? Everybody here know what E. coli is? Yeah. So it's the type of bacteria. The thing with E. coli, it replicates very quickly. So it can be used to study. So what they did, yeah, you all know what a culture petri dishes are. Culture dishes is like a petri dish, right? And it has some type of mixture in it, right? Like a jelly kind of mixture. What biologists do, they take a sample of the E. coli and they pass it on the petri dishes. What would happen if they leave it in the incubate, the E. coli would multiply. Right? So our first generation, second generation, and so forth.
That the fun factors, that's what I'm saying.
Yeah. So it have the kind of stripe
Yeah, it's example.
that is zigzag. So different types of bacteria culture in different. Or streak, it hasn't been stricken. That's you always learn about that in modern trick.
This, wow,
Right? So this is what they did, right? They had some E. coli and they stripped it or they plated it, right? On the petri dishes. No, they didn't plate it on a regular petri dish. So there's a petri dish. And they plated it on a special substance called nitrogen. It was 50. So in the petri dish, in the broth itself, right, in the substance it contains nitrogen 15. Another word for this is heavy nitrogen. Okay, so what they did, they cultured the E. coli, right? So the E. coli colonies grew on the petri dish. Okay, they kept reproducing, so they grew on it. Now these E. coli, their DNA strands would contain nitrogen 15. Right? Remember, nitrogen makes up the DNA. Remember the structure of the nucleotide pieces, right? It had nitrogen in it. So their DNA would contain nitrogen 15. What they did at the same time, the cultural next plate, but this time it contains nitrogen 14. Right, this is called light nitrogen. Now the reason this is called heavy nitrogen and light nitrogen, this represents the mass number of the nitrogens. Okay, so they are isotopes of each other. Okay, now if you look at the DNA under a spectrophotometer, you will see that this nitrogen is heavier than this nitrogen. So the E. coli from this colony would contain nitrogen 14 or light nitrogen. So what they did, they took some E. coli from this plate, they took some E. coli from this plate, and they put them on the same plate. Okay? So what they noticed after what? Sorry, they took some E. coli from this plate and they put it on this plate. Okay? Now, what type of nitrogen will E. coli, what nitrogen this DNA will contain? 50 or heavy nitrogen, right? When you put it on this plate now, what type of nitrogen you expect it to take on? Nitrogen 14, right? So all the E. coli on this plate would have DNA that looks like this. Nitrogen 15, nitrogen 50. All the DNA on when you take some of that and you put it here, you notice one strand is nitrogen 15, and the other strand add nitrogen 14. Yeah, because they took the nitrogen, the E. coli from this plate and they put it on this plate. The E. coli on this plate, when it was time to copy their DNA, they took off nitrogen from this plate. So if they took off nitrogen from this plate, only one of these strands had nitrogen 14. Okay, remember the originally started off like this. When they replicate, they start to replicate with nitrogen 14. They keep one of the original strands and they get a new nitrogen 14 strand. So that's how they prove that DNA replication is semi-conservative. If it wasn't semi-conservative, right? And the nitrogen 15 from here was placed in this petri dish, you'd expect that you get back the bases with nitrogen 15. Right? Because it would use it from you understand? Or the most. Guy understand? Sure. Say understand? Awesome. Yeah, it's something annoying or what to see a question and be like, what we going to do? All they're doing is remember DNA needs nitrogen, right? So where does it get nitrogen from? It gets the organism gets from the environment. So E. coli would have gotten nitrogen 15 from the petri dish. It's using nitrogen 15 in DNA replication. So all of its DNA originally would contain nitrogen 15. When you take some of the E. coli from this plate and you place it on here, when it was time for them to replicate their DNA, they took some of the nitrogen 14 from this plate. So one of these strands, the new strand, would contain nitrogen 14. Old strand would still have the nitrogen 15. Okay, that's what and this is what proven DNA replication is semi-conservative. Right?
But ever actually, all the months is connected.
From what that? From what? Oh, they'll answer. Will be from our diet or environment. I remember we eat proteins. Proteins is not nitrogen in it. But we don't use that nitrogen in our body. Remember, we can't use gaseous nitrogen. It's their diet too. Right? We just take a short note on this. Right, so evidence to support semi-conservative replication. Evidence to support semi-conservative replication. evidence to support semi-conservative replication. Cells of E. coli, cells of E. coli, E. coli were plated on a medium containing nitrogen 15, were plated on a medium containing nitrogen 50. Full stop. Some of these bacteria were taken from the first plate, and placed in a medium of nitrogen 40, and placed in a medium of nitrogen 40. Full stop. It was allowed to replicate for one generation. Was allowed to replicate for one generation. It was allowed to replicate for one generation. And it was observed, that one strand was the original. It was observed that one strand was the original nitrogen 15. The next strand was the original nitrogen 15. While the other strand contained nitrogen 40, and the other strand contained nitrogen 40. Full stop. So if it wasn't conservative again, after replication, these strands would just contain nitrogen 50. Okay, it'll be fully conservative. Right? If it was not conservative at all, the entire strand, the two strands would contain the new nitrogen 40. Right? That's it for DNA replication. Very good.
And then there's an inside in the axis of prime. You gotta really know what.
Yeah, so they're going to be in translation and transcription. So that's what we're going to get into now, right? Protein synthesis. Right, so that's it for DNA replication. Now we have to get into protein synthesis. No. It's usually in ChatGPT in the exam.
What? Yeah, we got because like it's more like we're not all dance productions. We can't take a chicken little big shelf side.
Oh,
Yeah,
I mean, it's,
you leave for like you leave right before she whatever.
well,
It's
like during school time.
you leave lunchtime.
Oh, yeah,
Oh yeah.
but she didn't even want to.
Yeah, well, yeah, I thought, you know, I told you the same of this, you know, the beach match. That's what.
But if it was like the children one.
Yeah, you trying to say,
No,
right.
say all these pictures was for me.
It was a couple big basket.
Right, the whole basket was for me.
Yeah, I think it was 4%. You got come for that.
I call it that.
Oh, it was people.
Yeah.
I'm so much classification. I'm following.
She was literally
Like, it was one night.
like this, and then I asked him to before the exam, I'm 40 seconds.
Gosh.
And then when I see her looking, I was like, you have your phone, she was like, no.
Well, and then she was like, then he went up, and I was like, put out the outside. And she think I was strawberry.
And you will say that too.
I only think that he was the cheat.
But was it sure so reality was cheating?
Yeah,
Okay, so-
No, no, but yeah, she took work from home.
Right, so we're doing protein synthesis, but before we get into protein synthesis, we have to talk about the genetic code, right? This would be a subhead. The genetic code. I'm not sure if the handles had this, so I'll just send you all it. Case
Oh, take it.
Yeah, Tim.
Because the code.
codes.
It's not like saying something. Like me,
It's just don't treat it.
gosh, it's not saying things.
That's the point of I.
And it just makes sense even to
Okay, so we know our genetic sequence, the DNA has 4 bases, right? What are those 4 bases? What are the 4 bases in the DNA?
No, it is how.
Well, you.
Right, so we have adenine, guanine, cytosine, and thymine, right? So these are 4 bases. Individually, we will call them a single code. Because it's just one base making up the code, right? If we were to combine them, so let's say we have let's say AT and CG, this will be a double codon, right? So it can also be EC, CA, these are double codons. If you put them in triples now, right, so these will be double codons, these are complementary to each other, they just next to each other. This is a double codon, this will be single codons. And if you put them in trees, there will be a triple codon. If we have something like TAC, this will be a triple codon. Now how these bases are read in our body is in triplets, right? In trees. So if you want, so if, let's say we have TAC, right? This TAC will code for a specific amino acid. Understand? So something passes along the DNA, it reads this, there's like, okay, this is the code for a certain amino acid. That amino acid will then be produced. Okay? How many amino acids are there again? 20. Right? So it have 20 amino acids. Anybody know from these four, right? How much can be possible triple combination start from these four?
64.
64. Anybody know how you arrive at 64?
Thank you.
4Q, right? So it has four bases. You print them in triplets. So that's how I get four. So 4Q will be 64. So now if you have 64 possible code sequences, right? And only 20 amino acids, what does that mean?
It's
It has different codons.
what's the same,
Wait.
or the same.
Right, so some of the codons would code for the same amino acid. What I just described here is something called a degenerate code. Means that some of the triple codons would code for the same amino acids. Right? Now, if you look at the table I sent you on the group chat, the first one. No, not the first one. Yes, the first one. Wait, that. No, you only require to name this right by heart. You have to know what to interpret it.
This one is.
So for you just type in the sentence.
So you could use look at it. It's happened.
It's not working. Right. Right, so if you look at the first one I sent, right? You'll see AAA and AAG codes for the same amino acid, PH. You'll see that? Right, so it simply means that this code is degenerate. Some of the codes going to produce the same amino acid. So anytime you ask you what is a degenerate code, you state that some different codons would code for the same amino acid. Good. Right. 64 triple. Okay, so our genetic code is degenerate. And it is non-overlapping. We will get into what this is. No, it's non-overlapping. Right. So, non-overlapping, generate. There's it. The two ones. Motion. Oh. And it's almost universal. So when I say it's universal, I mean almost all living organisms use this system. Right? It uses these four bases and it uses the same type of RNA. Okay? All living organisms, I should say, known living organisms. Right? Okay, so what do I mean by overlapping? So let's say we have a genetic sequence. CPT. G, E, G, E. So this is located on somewhere along the DNA strand, right? So there are two ways we can read it. If it's non-overlapping, we're reading it in triples. So what amino acid would this code for? C, so you can check the table. CTG codes for 8. Right? So asparagine. AGE. What does that code for?
Yeah.
SCR. So you're reading it in triples. You get that?
Yeah, CUG, right?
CUG. For this one? Oh no, you're looking at the first table. You already used for R. I'm looking at the one for K. Right? So CTG will be asparagine, AGA, SCR. These are the amino acids it's code for. And then the triplet next to it, you read it off like that. This is an example for non-overlapping. You have no overlap between bases. Right? When we talk about overlapping protein, it's the same thing. C, name after that, where you
Your fault.
C, T, G, A, G, E. a non-overlapping, sorry, an overlapping code. Let's say start from me. We know that produces ASP, right? The amino acid. However, it doesn't start where this one ends. So in non-overlapping, it will leave this one, go to HE. For overlapping, it will start in between this one. So it starts at T, so then it will go to TG. It has an overlap between the codes. One TGA code will THR. And then when it reads it again, it'll read it from G in a triplet to the other G. So it's GAG. You understand? This is an overlapping code. There's overlap between triplets or between codons.
Yeah.
Right? You all have to worry about this one because this is not how DNA works. Right? It works like in non-overlapping. It's not any difference. Right? So that's the difference, overlapping and non-overlapping code, you'll say. In a non-overlapping code, there's no overlapping or no sharing of bases. Okay? You just take down the main points, take down this as well. And this example. Yeah, or after they take down that. Right, on the same table as the first one, you'll see something that says stop, right? That simply means it's a stop codon. Right? A stop codon would tell whatever is doing the translating or transcription where to start. If it didn't have a stop codon, it would just keep on reoccurring. So you need a stop codon to tell to say, okay, yeah, let's stop at this point. Okay, so whenever it's stop there, it simply means it's a stop codon. It's not going to produce an amino acid. It's just going to stop at that point. We had stop in school. We had stop after mutations, right? But we didn't finish mutations, yeah.
Give you example of creation from evolution.
Okay. And so now we get into the protein synthesis parts. Anybody still taking on the board? Dr. Spread. Still taking on the board.
Say the something.
Is that one point?
Yeah, but you're giving me, no, what's story chat? Well, what's the sense something for you?
Yeah.
What else? I would say, yeah, then like, yeah, he would dress. Oh, not that, like, Prince William, so he needs to. I was talking about this, I don't think I'm just in there, like, I think I'm just probably doing this. I really don't think I was already. That's I think I'm really going to touch. She was coming back on. Because he went to culinary, I don't. Yeah. Yeah, I'm watching. It's gonna work. Yes, but it was actually, I was trying to remember that yesterday. Remember, I was like, maybe, I guess, I was like, then I was like, you have, yes, if I,
Right, so you have something called the central dogma of molecular biology, right? And it goes like, it starts off with the DNA, right? So we have everybody is born with DNA, right? Everybody has DNA. Your DNA is supposed to be constant throughout as long as you're alive. Okay? Now no changes. Your DNA, right, is then converted into RNA. So based on the DNA, that will be the RNA that you produce. The RNA is then used to produce proteins. Okay? Everybody have DNA. Your DNA is transcribed into RNA, and the RNA is translated into proteins. This is the central dogma of molecular biology. Right? Okay. Yeah, we're all 12 minutes.
Yeah, but transcription, translation.
Yeah. Right, so let's say we inside our nucleus, right? What would we find inside the nucleus? What do you mean we find inside our nucleus? DNA, right? So you have DNA inside your nucleus. Inside the nucleus, DNA. With DNA, right, is converted into something called mRNA. That M stands for messenger. This process is called transcription. So DNA in the nucleus is converted to messenger RNA by the process of transcription. The mRNA then leaves the nucleus, right, through the nuclear pores, and then it goes to a ribosome. Remember, a ribosome is involved in protein synthesis. So the mRNA goes towards your ribosome, and your ribosome, with the help of two other things, undergoes translation it undergoes translation to form a protein. Now, this protein could be enzymes, right? It could be proteins used to make hormones. It could be a set of different things, right? Proteins or polypeptides. Right, these polypeptides of proteins can then be used throughout the body. Depending on which type of polypeptide you need, that will be the ones that are produced. And they determine the characteristics of the organism. So when they say DNA determines the characteristics, this is how the DNA is responsible for the mRNA, which is responsible for the protein that's produced. So there's a simplified version of what we're doing, right? Right, so I said some things about the ribosome. Those things would be the tRNA, which stands for transport RNA, plus some amino acids. Right, you go towards the ribosome along with the mRNA, to undergo translation, forming a polypeptide. So take us on, it's a summary of the whole process. Again, it's a summary, missing some stuff. And next picture, and nobody telling me anything.
But
Very
that's
good.
really not sure, that's just like that.
A diary.
This is a diagram, isn't it?
And flow chart.
A flow chart. Okay, but the nucleus part is the drawing part.
I'm not horrible.
That's what you just said.
For a family, it's not down.
Yeah, but
Good job, good job.
let's see flow drawing from last week. It was all over the place. Compared to that, compared to.
Take pictures and put it on each other.
I shouldn't. Oh, let me see how they draw.
Oh.
No, let us.
My girl. One my turn. You see, that's
That's really cute.
What was
It's
this?
good.
Was that?
What was that?
What was that? What was that? What's this?
After like over last few people.
And I started.
So I would never do anything. I would always need something already.
Everyone was so, but you know.
What's the next one? Is that?
I so sure you got to be just that joy.
It's not like- So, you are part of my.
Yeah, but it should still be very bad. Like when big age you are compared to the loss.
I so wish I had this thing.
Now you're gonna move around.
Let's say you're back to like one person.
Alright, so you go to listen to this class, from what I could see, we tell me all the choices I know, right?
Like, chat, how many doors?
I have a good joy. But
Because
This should say that one, hey, I'm sketchy. Mine.
I have a joy. Like, get on this stuff.
You want this,
Oh, yeah, I felt too, you know.
you want this, you want to like sketchy.
no, well, it's
Hello, this.
why is that?
No.
Look how nice people say.
My CR.
Lovely. But now, I heard for my first workshop, greatness.
What's
Such
going on?
You're short.
He wasn't. If it's short, it's short, we'll try it.
Yes,
You skip the important function. Find out what to do.
I go back, I'm not a figure.
Oh, this is good.
What's your guessing? Sorry.
Oh, yeah.
Alright, so I think.
That-
I really don't like it. I really like it.
And look, like, look, that way you said you did it. Well, look, can I get this?
That was nice.
Okay, so was there two main processes involved in protein synthesis. Transcription and translation, right? They need data, and they'll come back after finishing the final, leave it. Right? Right. Transcription and translation, now both of them have substance, right? But the good thing is, it's the same substance for both of them.
It's different, it's mixed up. That's bad.
It makes sense.
That's bad.
Right? So in transcription, there are three parts. Initiation, elongation, and termination. Right? In translation, it's the same thing. Now the steps are not the same, right? But they just call the same thing. Right? Right, so I'm just going to briefly just go through it. It's to initiate means to start. So before, I asked me about something called primers, right? So your primers, when you think to that, those are like your start codons. So your entire RNA, the entire DNA sequence is not going to be converted into RNA. Right? Only certain parts that are needed. Remember, the majority of the DNA is not used. So only the parts in which the cell needs going to be transcribed to mRNA. How does it know which parts are needed? It has something called a start codon. Right? That start codon says, okay, it tells the enzyme start from this point. Right? And then it will start from that point and start to transcribe. It also has a stop codon. So the enzyme knows, okay, at this point I have to stop. Right? So that's initiation, finding that start codon and starting the process. Elongation is when you build up on it. So you produce your first amino acid, you have to produce the second, the third one, the fourth one, then join them together. You elongate and you lengthen them, the polypeptide, polypeptide. Termination is what I just talked about, with the stop codon. You have to stop at a certain point, you reach the stop codon, as it release the polypeptide into the cytoplasm. Okay? In next class, we'll go through these three steps for both transcription and translation.
It's the same steps.
No. Well, same name and steps. Still different.
But in it's more.
Right, so that's it for today. Anybody have any questions?
Why?
Any further questions? You need to go about it already further. We may know. Transcription, yes, it doesn't start first. Start, that's already. So, and you need to go with the translation.
Rather than looking into those is like hot. And you're saying that the they call it odd to one of the things, right? They will make the leader check. And they see the eye of the paper check. It's about the end.
Well, from here to here, and then it comes to the as well. And then it starts. But it starts with the. And you see this one.
Because in diagram, right, I was like, if I finish really know the thing is that, I just have to see it from the other side. But you should, right, there's like, I was like, oh, you know, next to it starts, so the top, which should be true, sure, yeah, but it was true, it was true, I was like.
If you want to take out the words.
Yeah. I don't know, this particular job was just like, stop, support it. I love the conversation.
You say, you're going to think,
I said, no, like, yeah, let me sketch it. I didn't like, you know. with this one, right?
Sure, you took only 4 years in the future. You, what were you also doing? Yeah, your picture.
Yeah, we did.
Because you can. Sorry. Right. So, yeah. So you think we start from here. Yes.
Start when you try. That's the beginning. Right? Start with here. Start with security. Or you can have like,
yeah.
yeah.
So if we'll start.
Yeah, but that's the beginning.
So
Yeah, you should have her.
So remember, it starts in the fight, if you come back.
Okay.
This one here, this is gonna be fine. That is usually, maybe that one kind of paragraph. If you want to shoot you know, it was a tool. Okay, now, show the copy, look at this, your product, it was what yours, which is so, it must be five. This was.
Yeah, there's a point. So,
Yeah. This is, you want to see, you know,
I get you. Because, then it was, I was surprised at it.
This.
Yeah.
Well, I need that for this. We have to be.
Okay, so. I mean, look at other ones, you know, like, seeing. Yeah. So there's the way you go through the slide. And I keep going back. It goes back. I'm not sure. And, yeah. No, I'll answer this question. Yeah, come on. Yeah, come on. Yeah, right, so, the, for the ways. Yes. Oh, this is good. Okay, I don't know. This is going to be right. Okay, so we did Shroud. Five. Release. 17. So, until it's not. The only reason why I kind of was like, I don't know if he's not like.
I'm trying to read this about 2 million. Oh, you know what? So, because I had this, they was leading show, you wanna leave copy, as a leading show. They have a deal as part, as the original movie.
Okay.
So it's the same thing, completely. It's
Almost so easy to understand with the.
yeah, but you want to, because of direction, so Gary will struggle, still go from tree to fire. Right, I had this label as tree to fire, tree to fire, and I this label as leadership, right? So this is fire, or this is tree. Okay, and this was the goal. Do the instrument still going tree to fire for the direction? So,
The channel goes through a fire and shoot them.
for a tree to fire.
Okay.
So, yeah, we need to try, there's no tree to fire.
Okay. I know that's how it is.
So I just had tables. So in my one, you need the shrine was the copy part.
Okay.
Name of the shrine is the original. But it means simple. Was the direction this sense?
Because there's nothing else, obviously not online, it's always in school.
Oh,
So that's why I was like, a little bit of why was the opposite me? Give me out. I think
but it's like the same thing, it's just like, okay, if you want to compare something, you get the same image, but actually means it's it.
okay,
So it's like that.
understood. Alright, take care, sir.
Yeah, it's